Chapter 14, Section 4, Mutations. Learning objectives: describe how mutations change genetic information. Explain how mutations affect genes. Now and then cells make mistakes in copying their own DNA. An incorrect base is inserted, or a base is skipped during transcription. These variations are called mutations, from the Latin word mutere, meaning to change. Mutations are heritable changes in genetic information. Mutations can involve changes in the sequence of nucleotides in DNA or changes in the number or structure of chromosomes. There are two different types: gene mutations and chromosomal mutations. Mutations that change a single base pair are known as point mutations because they occur at a single point in the DNA sequence. Point mutations usually involve a substitution, in which one base is changed to a different base. Substitutions usually affect no more than a single amino acid, and sometimes have no effect at all. Mutations that change the amino acids specified by a codon are called missense mutations. Sometimes a point mutation can have severe effects on gene expression. This is known as a nonsense mutation because it would cause translation to stop before the protein was finished. Depending on the position of the stop codon, it could result in a production of a defective protein. There are three different types of point mutations: a substitution, insertion, and a deletion. Mutations in which one or many bases are involved or removed from the DNA sequence are called insertion and deletions. The significance of these changes can be dramatic. Remember that the genetic code is read 3 bases at a time. If a nucleotide is added or deleted, the bases are still read in groups of 3, but now these groupings change in every codon that follows the mutation. Insertions and deletions are also called frame shift mutations, because they shift the reading frame of the genetic message. By the shifting of the reading frame, frameshift mutations can change every amino acid that follows the point of the mutation. They can alter a protein so much that it is unable to perform its normal functions. Chromosomal mutations involve changes in the number or structure of a chromosome. These mutations can change the location of genes on a chromosome and can even change the number of copies of some genes. There are 4 different types of chromosomal mutations: deletions, duplications, inversions, and translocations. Deletion involves the loss of all or part of a chromosome. Duplication produces an extra copy of all or part of a chromosome. And inversion reverses the direction of parts of a chromosome. Translocation occurs when part of one chromosome breaks off and attaches to another. Genetic material can be altered by natural events or by artificial means. The resulting mutations may or may not affect an organism, and some mutations that affect individual organisms can also affect a species, or even the entire ecosystem. Many mutations are produced by errors in genetic processes. For example, some point mutations are caused by errors during DNA replication. The cellular machinery that replicates DNA inserts an incorrect base roughly once every 10 million bases, but small changes in genes can gradually accumulate over time. Some mutations arise from mutagens, chemical or physical agents in the environment. Chemical mutagens include certain pesticides, a few natural plant alkaloids, tobacco smoke, and environmental pollutants. Physical mutagens include some forms of electromagnetic radiation, such as ultraviolet light, and X-rays. If these agents interact with DNA, they can produce mutagens at high rates. The effects of mutations on genes vary widely. Some have little or no effect. Some produce beneficial variations, and some negatively disrupt gene function. Some of the most harmful mutations are those that dramatically change protein structure or gene activity. The defective proteins produced by these mutations can disrupt normal biological activities and result in genetic disorders. Some of the variation produced by mutations can be highly advantageous to an organism or species. Mutations often produce proteins with new or altered function that can be useful to organisms in different or changing environments. Plant and animal breeders often make use of good mutations. For example, when a complete set of chromosomes fails to separate during meiosis, the gametes that result may produce a triploid or 3N, or even a tetraploid or 4N in organisms. The condition in which an organism has extra sets of chromosomes is called a polypeploid. Polypeploid plants are often larger and stronger than diploid plants. Important crop plants, including bananas, limes, and strawberries, have been produced this way. Polypeploid also occurs naturally in citrus plants, often through spontaneous mutations.